Thanks so much for the introduction. I hope you can see the slides.
Yes, we can see well.
Perfect. So yes, my talk, as Xiao introduced, is on machine learning on sequence and structural data. So I will show several projects of the lab. So I will not try to go too much in depth, just give an overview of what we're doing right now. And I would like to mention, first of all, that most of the work that I'm showing is in very close collaboration with Gerhard Sandwe, who is a professor at our university as well, at the informatics department. So in a review that we published in 2019 in Molecular Systems Design and Engineering, we mapped out several progress and challenges of the AIRR-seq field with regard to genomics, proteomics, computational immunology, and biotechnology. And although there have been key advances in the past decade, I would say, the outstanding challenges that remain, I think, are so one of the main challenges, I think, is prediction of antibody-antigen binding or T-cell receptor antigen binding, and especially with regard to that, also the machine learning analysis of immune receptor repertoire. So I will focus on that in my talk. So machine learning on AIRR-seq data can be seen from the repertoire level. So I have this little icon here where we want to predict the class of a given individual. So we want to predict based on the repertoire, the patient status, so health, disease, or different diseases. And the problem with this is that from most of our repertoire data, as also shown in the previous talks, we don't have the antigen binding information. But what we want to find using machine learning is to find those disease-associated sequences that predict then the disease status of the individual. And I'm saying here disease-associated because it could be that those disease-associated sequences are not actually antigen binding. So it doesn't have to be a one-to-one relationship. And it's, of course, a very difficult problem to find those sequences if you do not know what they bind. So you kind of have to look at subsequence overlap, clonal overlap, and the machine learning approach has to kind of learn this. And in the machine learning field, we call this problem multiple instance learning problem, where you have several bags of sequences and only a few of them predict the status of the individual, and all the others can be seen as pseudo noise. On the sequence level, the machine learning problem that the AIRR-seq field has is the prediction of immune receptor antigen binding. Here I show antibody antigen binding, and especially the prediction of where the paratope is and where the epitope is. And this is especially important, of course, for epitope prediction. We want to know where the T-cell receptor or antibody binds on the antigen, or if it binds to the antigen, and then where it binds on the antigen. And then from a vaccine design point of view, that's very interesting, because if we were able to predict binding, then we could also know how to change antibody surface or protein surface to better to make vaccines more efficacious. We have recently also summarized the recent progress in the machine learning field in the review that just came out in Current Opinion in Systems Biology, where I just tried to summarize quickly again what I just said. So we have the sequence classification task, where given a sequence, we would like to predict binding or non-binding, or something in between. We have the repertoire-based classification, where we would like to predict based on a bag of sequences whether a person is healthy or diseased, and then only a few sequences actually predict disease, or so disease 1 or 2, or health or disease. And then rather new approach is also to be able to generate new sequences based on a given set of, for example, antigen-specific sequences, that you would like to make more of those antigen binding sequences with a different sequence but same antigen binding. And this is very nice for monoclonal antibody discovery, for example. The problems with machine learning that we have in general, we have summarized in this image. And here it is the low clonal overlap across individuals. So many individuals are very unique. So how do you find a common frame of reference? We have influencing factors that influence health or disease state. Mostly the disease signal is very low in the repertoire. We have poorly reactivity, so many-to-many networks. So a given immune receptor can bind to many antigens, and a given antigen can be bound by many different immune receptors. The immune signal is distributed across the frequency space, the sequence space, and the structural space. And then, as I mentioned, there are these factors that could be potential confounding factors. So for example, we know that age changes repertoire. So when you have only, for example, young individuals for a type 1D study, and you compare them to old people, then you have this age factor, for example. And then, of course, you have also technological factors that we still need to better understand how they influence computational and machine learning analysis of immune repertoires. So what has been done so far is that with respect to machine learning approaches, there are simulation data sets, simulation frameworks that allow the sequence generation of large data sets so we can benchmark machine learning approaches. Several studies have been shown and published that show that it is possible to predict based on the repertoire disease state. Several studies are going into the direction of TCR or antibody antigen binding prediction. And then, as I also already mentioned, there are several studies that begin to understand how we can use generative machine learning for making novel repertoires and sequences for maybe therapeutic purposes. So the first thing that I would like to present is so now I told you before that there are all of these machine learning approaches, but the problem for now in the AIRR-seq field is that many of those approaches are non-standardized. People sometimes reinvent the wheel. So it would be nice to put this all into one software platform where it's possible to reuse code from different groups, which allows flexible implementation of machine learning approaches, and that also allows the development of machine learning approaches that are adapted to the structure of AIRR-seq data. And this is this is what is currently captured in a platform that we are developing, which is called ImmuneML, for which we also have a poster and software demonstration at the AIRR-C meeting in a few days. So how does ImmuneML work? We have, you can, it encompasses all the steps that you would like. So dataset pre-processing, so it reads in your pre-processed data, it encodes the data. You can then apply different machine learning approaches, and then it allows exploratory analysis of the machine learning models and any other things that you would like to have. And everything is guided by a text file in which you can put your data analysis specifications in a very specific and reproducible way. And what it allows is then a real user friendliness, and it's easy to add new functionalities. It's modular, all of these things. The nice thing is that it can be applied to more biology-focused questions, such as we've seen before, but also more machine learning-guided questions for benchmarking of new machine learning approaches. And it's not only command line based, it's also galaxy based. So also for kind of people that come more from the biology field that don't have so much programming experience. Also for those people, this framework can be used. In this framework, you can also then use a deep learning approach that we have previously published with the Günther Klambauer lab in Linz, Austria, which is an attention-based deep learning approach, which allows repertoire-based classification and especially addresses this problem of multiple instance learning. So trying to find those sequences out of a bag of sequences that actually predict immune state. So we benchmark this towards the CMV study that was published in 2017, where those CMV specific clones have been found that predict CMV status. And we could find those back perfectly with our deep learning approach. And we have also benchmarked this on simulated data in this approach. So this is also free to use. It's on GitHub as shown here. And it's recently been accepted at NeurIPS. So we have also published this year a simulation framework for immune repertoire data, where you can generate your own data sets and then know exactly how they have been simulated, which is very important for benchmarking approaches where you want to find different patterns in your data. And we are now expanding this framework where we are starting from a repertoire generator such as ImmuneSim or AIRR to make it a really machine learning friendly simulation framework where you can have many different classes, where you can then implant many different motifs, where you can then also test whether those motifs are actually destroying the sequence structure of the sequence. So with respect to generation probability, et cetera, et cetera, et cetera. And this is ongoing work, and there's also a poster at the conference. So to summarize, we are currently developing and hopefully putting out soon a preprint on the ImmuneML framework, which allows novice to expert operated ML approaches on AIRR-seq data. We have published a attention-based deep learning approach for multiple instance learning for repertoire analysis. And we are very much interested in developing simulation frameworks to actually benchmark existing and new machine learning approaches. So now I'm coming to the third part of my talk, which is on antibody antigen binding prediction, which we are very interested in. And I think it's one of the longest standing questions in the field. If we can predict antibody antigen binding, and if yes, to what accuracy? With what accuracy? And this is based on a preprint that has been out for a year now, so I will go quite quickly so I can go to the new research that we have going on in this field. So what we have done in this paper that is, as I said, on bioRxiv, it's Akbar et al., so it's shown here. We have curated one of the largest data sets of antibody antigen crystal structures, so 825. So we took them from a curated database where we mapped the antibody antigen binding site, and especially the paratopes and the epitopes, which are shown here in this pink letters. And then we just first of all checked whether the paratope actually always maps to the CDRs, and they do. So only 10% of antibody residues that are in contact with antigen go to the framework. 90% is in the CDRs. And to better understand the binding patterns that are apparent across antibody antigen crystal structures, we devised a motif language, which is shown here. I'm not going too much into detail, but we basically showed that these motifs are very specific to epitopes and paratopes, so there's low overlap. We showed that we have discovered a large extent of them already, given that they're also quite simple. Yes, so basically, in our data set that we have now, 50% we have already discovered on the paratop side and about 15% on the epitope side. And we also showed that these motifs actually capture underlying sequence dependencies and thus biological meaning, have thus biological meaning. What we then did, and this is one of the most important graphs of the paper, is that we connected paratope motifs to epitope motifs. And we could do this because of this motif language that we came up with. And what you see is that we get a tree-like structure. So there are so specific paratope motifs in blue like to only connect or bind to a certain set of epitope motifs. So you have mutually exclusive polyreactivity, and given this tree structure, we can already, just from this tree structure, without actually saying anything about machine learning, we can already say that a priori paratope-epitope binding is predictable. And we checked via randomizing this framework, this network, that this structure that we see here is not just based on the data, but is actually true biology. We also checked that non-immune protein binding does not look like this. This is also in the paper. I'm not showing this here. And then we applied machine learning to these motifs, and we showed that they are the lower the number here is, the higher the accuracy. In pink, you see the random control, and in blue is the true data. And you see that if we are on the motif level, we can predict much better than random. If we go to the sequence level, so the true, where we want to really predict paratope-epitope binding, you see that accuracy is not really good yet. So we need more data, we think. But what we see is that if we pair sequence encoding with our structural interaction motifs, then the prediction accuracy gets slightly better. So hinting to the fact that structural information helps with prediction. So what we have shown in this paper is that there is a priori predictability of antibody-antigen binding. However, we have a data problem. We do not have enough antibody-antigen crystal structures to actually go more into structural prediction testing of machine learning approaches. So in order to resolve this problem, we are currently finishing up a preprint also for a simulation framework for a 3D antibody-antigen binding. And that we also have a poster at the meeting about this. And what this is this in the 3D framework, which is on a lattice base, we take PDB antigens, we encode them in a 90-degree lattice, and then let antibodies here in green bind to it, and then calculate the affinity and the energy. So we do this in all multiple billions of ways that this could happen, and we select the lowest energy binding. What this then gives us is that for each for each antigen that we encoded here like this, we get a 3D structure with paratope-epitope information and affinity, which is great for benchmarking machine learning approaches. So we have now here a database with 1 billion synthetic antibody-antigen structures, which took quite a long time to compute as we have to go to all of these bindings. Of course, our binding is not the same as the real binding, because if our in silico binding would be the same as the real binding, that we would have the Nobel Prize. So but still, our discretization of the protein into this lattice framework is in a 3 to 4 angstrom precision, so it's quite good. We take for the energy, so for the affinity calculation, we take real amino acid interaction potentials as measured by experimental data. We get the same features that we would get from crystal structures. And if we look at these interaction networks, here is again the one that we see from our experimental data, the 3D experimental data that I just showed you. And our interaction networks also look quite similar. So you also see this tree-like structure coming up here, just like here also. So that's very nice. And this is based on 7 million sequences. And here we have 800 sequences. Yes, and what you then can do with this framework is then, of course, you can investigate different machine learning approaches with respect to the different antigens. So I'm not going into detail here, but then you can look for each antigen, how the prediction accuracy is. You can look whether structural information helps prediction accuracy. You can define binders based on top affinity, top 1% affinity, top 5% affinity, and so on and so forth. So it's really nice, and we are currently exploring many different things with that. In the last part of my talks, I'm on time, so that's good. We wanted to also check whether actually generative machine learning preserves antigen binding, which we can only do in the simulation framework, which we cannot do experimentally, because which I will tell you now. So we took this simulation framework that I just showed you. We took high affinity antibodies towards 10 different antigens, and for each of these high affinity antibody sequences, we have the binding affinity, the paratope-epitope interface, and also different developability parameters. We put them into a recurrent neural network. Let them, so we put only the antibody sequences, so no 3D information, no actual antigen or paratope information. We make new sequences using a standard recurrent neural network, and then in our simulation framework test again the binding of these newly generated sequences, for which we then again get affinity, paratope, and epitope, and then we can look whether we find actually the same epitopes again. So what we did, and this is what we did. So we had 10 different PDB antigens. These are the identifiers of these PDB structures. And this is how the training, so blue is training and yellow is what comes out. And you see that yellow and blue sequences have the same affinity, but the yellow sequences are completely novel. So they are completely different from the blue training data set. But the affinity is the same as in the training data set, meaning we can make, this is a proof, an in silico proof of principle that machine learning can, based on 1D sequence data, if you have enough of this data, make new sequences that bind again the same target with epitope specificity, which we show here. So we find the same so we find similar paratopes and similar epitopes. We find even more epitopes, so you can find more different epitopes also with generative learning. We then also checked the developability. So there are different developability parameters which we checked, and we get a broad distribution of developability parameters, which is of course very nice for pharma companies to know that machine learning gives you actually a broader scope of physical chemical properties to go forward with your pipeline. Yes. So just to summarize again, what we showed is that we have now a simulation framework which can simulate synthetic structural antibody antigen binding at super high throughput. We can use this, which I didn't go into detail, to really now formalize machine learning based prediction of antigen binding, because most of the time when people say we want to predict antigen binding, it's not really clear what this means. And with this framework, we can actually formalize this in a machine learning task. And all of this allows us basically unconstrained in silico exploration of antibody specificity prediction. Using this framework, we have already provided an in silico proof of principle that generative machine learning can make epitope level specificity with epitope level precision new antibody sequences. And I didn't show you that you can also use transfer learning. So we also already benchmarked transfer learning. So you can take from one antigen, you can take this network and use it for to a certain extent for the generation of sequences for another antigen. And we hope that this work kind of paves the way actually, or gives trust to this generative machine learning field, that we can, in the future, given enough experimental data, design fit for purpose antibodies. And with that, I would like to come to an end and acknowledge my lab members, the group of Gerhard de Sandra, our funders, and all of our collaborators in China, Switzerland, the US, in Austria, and Switzerland. Yes, thank you very much for your attention.
Thank you, Victor. Very nice talk. Any questions? So before somebody you raise the question, so I think I noticed that after you prediction of your paratope and epitope specificity, you use some of the natural data to validate the feasibility of this prediction. So did that those antibody specificity being used to train your framework, or just leave for validation?
That's a very good question. So we haven't used. So if I go back here again, so we have kind of. So we have some kind of. Yes, experimental. Yeah, no, yeah. So yeah, so it's only after the fact, basically. So it's not. So of course we take. So we have some kind of biological reality in our simulation frameworks, such as those potentials here, for example, or we take real PDB structures and then put them into this discretized framework. But using anything from the previous work in this work we haven't done so far, and that's actually also something that we're looking into. Yes, yeah. So for now it's only for validation, but I think in the future, I think we don't have enough 3D antibody antigen structures yet from crystal structures to really put a lot of information in the simulation framework. What we did though is from the current data is to really check that our simulation framework gives us the same features, at least, as the crystal structures. Yeah.
Yeah, I agree. So in the future, maybe for repertoire database, we should also include the structured data of the
So, and the nice thing is here that here we didn't use any structural data for the training. So here we just used 1D sequence data, and then we showed that basically the 3D structural dependencies that are important for binding the antigen are learned by the machine learning approach. So basically what this work here says is that we maybe don't need so much structural data. What we need is large scale antigen specific, maybe phage display data or something like that to really train the machine learning approach as well. Yeah, and that is much easier to get than large scale structural data. Yeah.
Thank you.
Yes.